this video, we are going to learn about the subtopic of chapter 22 in Meader textbooks, which is evolution and characteristic of fungi. Have you ever wondered about the evolution of fungi, the structure of fungi, and how fungi can reproduce? Well, let's learn about the curiosities in our video. Fungi were the first eukaryote that invade land. They are ancient organisms with deep underground networks of mycelium or fungal roots that service the ecosystem by recycling organic debris back into topsoil. The part of the fungi we see and sometimes we eat is the fruiting body, like a mushroom, which is the center of reproduction for many species. The medicinal properties of fungi have been known for centuries. These fungi have antimicrobial and antiviral properties. In many cultures, fungi play an important role in non-traditional medicine knowledge, which is now being tapped by pharmaceutical research. For example, many species of fungi are entomopathogenic, meaning that they specifically attack insects and insect larvae. The cordyceps fungi produce spores that attach onto caterpillars. The spore germinate, then digest and kill the caterpillar with fungal hyphae. The spore-producing fruiting body will later emerge from the head. Caterpillars with cordyceps fruiting body are an expensive Chinese medicine used to treat respiratory, kidney and liver disease. Fungi that attack insects are considered environmentally safe, and there is a growing interest worldwide to use fungi as a biological control against insect pests such as mosquitoes. Products such as antibiotics, antivirus, and biological control derive from the natural processes fungi. It has the potential to revolutionize human society and our impact on the environment.
Now I want to continue with evolution of fungi. Recently, there are 6 phyla are recognized, and there are still many want to define. Animals and fungi also both in the supergroup Epistoconta, which means they are related to each other than plants. Animals and fungi is actually both like the same because they are both heterotroph, which means they can produce their own food, and fungi is saprotroph, which means they can absorb nutrients from their surroundings. Next, fungi does not fossilize well, so it is difficult to estimate when is the fossil record started from.
Okay, so next I will explain more about the structure of fungi. So this is our 3D model that we made. We choose a type of fungi which is septate hyphae. It consists of nucleus, septum, hyphae, cytoplasm, and cell wall. So some fungi, including the yeast, are single-cell organisms. However, most species are multicellular. Those multicellular fungi have a structure known as a mycelium. A mycelium is a network of fungal filaments which has a thin structure called hyphae. Hyphae give the mycelium quite a large surface to volume ratio, and this maximize the absorption of nutrients into the body of a fungus. It is also either septate or aseptate fungus. So what are aseptate or anaseptate fungus? Septate fungus is fungi that have septa, which are the walls of tissue in their hyphae, and these septa or septum are formed behind the growing tip of hyphae. Therefore, this will cause the partitioning of the hyphae into individual cells. So as we can see from the 3D model, the hyphae is divided into individual cells because the presence of septum. So whereas aseptate fungus is fungi that do not have septa. Septa also have pores in it. So that pores in the septa allow cytoplasm, and sometimes even organelles to pass freely from one cell to another. Receptor that separate reproductive cells, however, are completely closed in all fungal groups.
As mentioned by ALYA just now, there are two types of fungi which are septate fungi and aseptate fungi. In most fungi, hyphae are divided into cells by internal cross walls called septa. However, some fungi have aseptate fungi, meaning that their hyphae are not partitioned by septa. These aseptate fungi are not divided into cells, and many nuclei are present in the cytoplasm of a single hypha. Some hyphae are capable of penetrating rigid substances such as plant tissues. When a fungus reproduces, a specific portion of the mycelium becomes a reproductive structure which is nourished by the rest of the mycelium. Next, we move to the differences between fungal cells and plant cells. Fungal cells are quite different from plant cells in which they lack of chloroplast and have a cell wall made of chitin. Chitin, like cellulose, is a polymer of glucose organized into microfibrils, but each glucose molecule of chitin has a nitrogen-containing amino attached to it. Chitin can also be found in the exoskeleton of arthropods, which is a major phylum of animals that includes the insects and crustaceans. Apart from that, unlike plant cells that store energy as starch, fungi store energy as glycogen, the same molecule that animals use to store energy. However, fungi are not motile and do not have any flagella at any stage in their life cycle. Therefore, they move toward a food source by hyphae growing toward it. And for your information, growing hyphae can cover as much as a kilometer a day. Amazing, isn't it?
Now, I'm going to talk about the reproduction of fungus. Some fungus can reproduce in both sexually and asexually. In the life cycle of sexually reproducing fungus, a haploid phase alternates with a diploid phase. In the haploid phase, hyphae from two different parents make contact and fuse. In some species, the nuclei from each parent fuse immediately, while other species do not fuse for days, months, and years. Before fusing, the nuclei continue to divide, in which that every cell in septate hyphae contains at least one of each nucleus. So, a hypha with paired haploid nuclei is said to be dikaryotic or N+N. Then, when the nuclei finally fuse, the zygote undergoes meiosis, resulting in the formation of spores. In the majority of fungi, all structures are haploid, except the zygote. Spore is a reproductive cell capable of developing into a new individual without fusion with another reproductive cell. To elucidate, spores differ from gametes, which are reproductive cells that must fuse in pairs in order to give rise to a new individual. Usually, fungi produce non-motile spores. For example, a large mushroom can produce and release billions of spores in a few days through the rainfall, wind, and insects. When the spore lands on an appropriate food source, it germinates and grows. Plasmogamy is a stage in the sexual reproduction of fungi in which the protoplasm of two parent cells from the mycelia fuses together without the fusion of nuclei, effectively bringing two haploid nuclei close together in the same cell. Karyogamy is the process that results in the fusion of these haploid nuclei and the formation of a diploid nucleus, which is a nucleus containing two sets of chromosomes, one from each parent. The cell formed by karyogamy is called the zygote. Next, fungi reproduce asexually by producing spores, fragmentation, and budding. The most common mode of asexual reproduction is through the formation of asexual spores by a single mycelium. For example, spores that produce asexually by a mushroom are produced by one parent and are genetically identical to the parent. The spores land to the ground and develop into new fungi. Fragmentation is a form of cloning. For example, moths produce tiny filaments called hyphae. These hyphae obtain food and nutrients from the body of other organisms to grow and fertilize. Then, a piece of hyphae breaks off and grows into a new individual, and the cycle continues.
Buds are produced from almost any point of the fungi parts. Just like yeast, a tiny bud gets pinched off and it will grow to the full size. The bud eventually develops into a new fungi, duplicating the parents.